All right. So thanks for the introduction, and thank you all for coming. So the optical microscope and optical telescope were developed contemporaneously about 400 years ago. And immediately they started to have an impact in revealing new worlds above our heads, but also new micro worlds all around us. For the next 400 years, the telescope steadily improved as it got bigger and bigger apertures, collected more and more light, got better resolution up to a point. And today, of course, we can see right to the very beginnings of the universe at about 13 billion light years away. The optical microscope also got off to a fast start, in part because of Antony von Leeuwenhoek. And he was able, by the 1680s, even to be able to see individual bacteria. However, von Leeuwenhoek's idea was something he kept to himself, so that when he died, it was 200 years before anybody could make a microscope as good as his. And so finally, in the late 19th century, engineer Carl Zeiss, physicist Ernst Abbe, and chemist Otto Schott teamed up together to finally make a microscope that could beat von Leeuwenhoek's by about a factor of 4. But they also found that there was a limit to how fine you could see, and it's roughly equal to half of the wavelength of the light you use at the highest apertures that you can create. And so that's where things stood. And in fact, by 1980, the microscopes you would buy at that time were actually very little different in terms of resolution performance than the microscopes you could have bought from Zeiss in 1880. Basically, things stalled. Whereas in telescopes, of course, there was a huge advance throughout the 20th century. So things changed around 1980 because of 3 technologies that came together. The first was, of course, the transistor and integrated circuits, which led to widespread computers that were cheap, that could run experiments, analyze data, and likewise led to imaging sensors that were very sensitive, like CCD cameras and so forth. The next innovation was the laser, developed in the early 60s, which then allowed us to be able to use very pure colors of light and coherence to be able to control the phase of the light, which was important in microscopy. And the third thing was, even though it was 100 years old, the idea of getting contrast by fluorescence as opposed to by bright field imaging or anything else turned out to be very powerful in conjunction with lasers, because now you could specifically label specific proteins that you wanted to see inside of the cell or specific organelles and get only those to light up. And so it provided this contrast that was difficult to get by other means. I have a love-hate relationship with fluorescence, but its main advantage is it's simple, it has single molecule sensitivity and tremendous specificity. And with the invention of green fluorescent protein, where you snip some DNA out of a jellyfish and splice it into any organism you want, you can get any protein you want to glow inside of a living organism. And all of that helped to push optical microscopy along. So I was lucky enough to get into this field just as these things were coalescing. And the first 2 microscopes that were created based on this were the confocal microscope and the 2-photon microscope. And both those instruments have had an enormous impact on biology. My interest was, thanks to my advisors, to get around that limit that Abbe described on resolution. And the idea we had was to create a sharp tip like you see there that has a small hole on the end of it, which is much smaller than the wavelength of light. And if we shine light through that hole, then it will act as a little nano flashlight, which we can drive point by point across the specimen to beat the diffraction limit. So that was the idea. After about 6 years of work in graduate school, we poked a little bit beyond the diffraction limit with that technique. That's what we call near-field microscopy. And then that was good enough to get me in the door at Bell, and then I had my own lab as an MTS for 6 years, in which we refined the technology and started doing applications. At one point, we had the world record for data storage density. We were able to do the first fluorescence imaging of cells that you see at the right there when we're looking at the actin cytoskeleton. I was the first person to be able to see single molecules that you can see at bottom left at room temperature. And with my buddy Harold, we were able to study the local nanoscale spectroscopy of quantum confined structures, like quantum wells like you'll find in a laser pointer. But after now 6 years of grad school and 6 years at Bell, I was very disillusioned with the whole field. The first source of disillusionment was not a good enough gut appreciation of the Achilles heel of the method, which is that the light that comes out of that small hole spreads extremely rapidly. And so if you're more than about 25 nanometers away from the sample, you get a significant loss of resolution. Well, the reason I got into this business back in '82 was I wanted to make an optical microscope with the resolution of an electron microscope to look at living cells. And it was clear that a cell's a lot rougher than 25 nanometers and a lot thicker as well, so this was not going to be the path to glory in order to do that. So what I've learned, basically, was my first lesson in a hard thing to learn, which is, as a technology developer, I've worked on probably about 6 different technologies, and they all follow the same trajectory in that I think when I get into it, I devote every ounce of my effort, put all the blood, sweat, and tears into it. And so you end up loving it like you love your own babies. And just like you think your baby is going to cure cancer or become president, in the end, you're just happy if it stays out of jail, okay? And that's the way it is with these technologies, too. Because basically, you get in when nobody believes in it, you start to gain some traction and see that you're on a good trajectory, and you start going up. My expectation is the yellow curve. When I realize what all the limitations of the technology are, you asymptote to the red curve. The other problem, though, is that when everybody else starts to see the applications I just described, the field gets extremely crowded with people. Most of those people don't know what they're doing. And so what happens is the signal stays about the same and the noise goes to infinity, so the SNR drops to 0. Okay? And so I found that very frustrating, in addition to being frustrated over the physical constraints and feeling that, it was probably not time well spent. I really felt like every good paper that I did just was the justification for 100 pieces of crap to follow in its wake. And everything I did was a net negative to society because it was a waste of time and the taxpayers' money. And so I dropped out of Bell and I dropped out of science. And a few months later, when I was pushing my daughter in a stroller, it occurred to me that I could combine my single molecule experiment with the quantum well experiment I did with my buddy Harold to come up with a different way of doing superresolution. So again, for biologists, by far the most important mechanism is fluorescence. And fluorescence in the end comes down to looking at discrete molecules that are tagging the thing you really care about, which is the native proteins they're linked to. And the problem is in a regular optical microscope, each one of those molecules makes a big fuzzy round blob. And those fuzzy round blobs run together like you see at upper left. But one of the things we learned in that quantum well experiment is that we could still see discrete sites of emission underneath the same near-field aperture, even though they weren't resolvable because they were all under the aperture at the same time. They glowed in different colors, so we could interrogate them based on their different colors individually. And so you can build up a higher dimensional space to look at molecules one at a time. So if we can do that, then those fuzzy blobs become fuzzy spheres, and then we can isolate, and once they're isolated, we can find the center of this fuzzy sphere to much better precision than its diameter. And so, as it turns out, you only need a few hundred photons to get 10 nanometer precision on the locations of those molecules. If you could do that with all the molecules, you got your molecular image. The problem is there was no good technology in 1995 when I pitched this to be able to isolate what might be hundreds or thousands of molecules in one diffraction limited spot. So I just published the idea and let it lie. And in the end, I worked for my dad's machine tool company in Michigan, thinking that as a good physicist, I could single-handedly save the Rust Belt from the Japanese, which showed how ignorant and naive I was. And in fact, my technology that I developed, although I was proud of it, was a complete disaster and burned through over $1 million of my dad's money. And so by 2003, I was unemployed again and trying to figure out what to do. I decided I wanted to get back into science for various reasons. I was motivated in part because of reading about that green fluorescent protein, which appeared 10 years earlier, but I had missed the boat because I had turned my back on science. But when I learned about it, I knew it was really important, and I had to do microscopy again. And so I came up with an idea for a microscope, which we'll talk about later. But in trying to pitch that microscope, I reconnected with my buddy Harold, and I tried to get him to do it with me. And he wasn't really interested, but he was going to help me try to find a home for my idea. And so one of the places we visited was Florida State, where we learned about a new type of fluorescent protein. You see it upper right, where if you shine blue light on it, nothing happens. Whereas in a normal fluorescent protein that you see at the right side, if you shine blue light on it, it glows green. But if you shine violet light on it, you activate the fluorescent proteins, and then the blue light makes it glow green. And so Harold and I realized this was the missing link to make that idea I pitched in '95 to work, because if we can turn on the violet light really low, we can turn on just a couple molecules at a time. They're then likely to be separated from one another far enough that we can find the centers of the fuzzy blobs accurately. And if we just take many, many images of different subsets of those molecules, we can build up the super resolution image. So I had convinced him some months earlier to quit his job and also try to find a way back into science. And so because we were both unemployed, the easiest and quickest path to getting this done was doing it ourselves. So we built the instrument in his living room, and that's now, I guess, an iconic picture as a result. So we needed biological help, so I was able to connect to the inventors of that photoactivated fluorescent protein, Jennifer Lippincott-Schwartz and George Patterson. And within a month of bringing the microscope there, we were taking little thin sections, about 80 nanometers thick, through a cell to look, in this case, at lysosomes. This is some of the initial data showing the molecules turning on at left, building up a diffraction-limited image by just summing the fuzzy blobs in the middle. But on the right, you're seeing something else building up by plotting the positions of the molecules. And after 20,000 images, you go from the left there to the right, or to better appreciate the resolution gain from that to that. So basically, with good enough samples with high enough density labeling, and we'll talk more about that in a minute, you can pretty easily get down to about 20 nanometer resolution, or 10 times beyond the diffraction limit. And with a technique that's simple enough that you can do it in your living room. So as a result, this became huge overnight, and the whole field just blossomed poof like that, and it got me the Nobel Prize. So for the next couple of years, we lived and breathed Palm by another crazy set of circumstances that again involved connections to Bell. My ability to get back into science was totally based on my time at Bell and having connections everywhere I could call on in my time of need and have people that would vouch for me. And so Harold and I ended up both at working for Howard Hughes Medical Institute in the new Genelia campus. And so this is some of the applications at top. We were working with Jan Liphart and Ed Wingreen looking at chemotaxis receptors in E. coli. We're able to show that the sizes and positions of those clusters you see of these chemotaxis receptors is completely predictable in terms of a stochastic model of self-assembly, where they insert in the membrane randomly and diffuse around until they stick. The middle one shows work with Bob Tejan at Genelia, where we're able to show that one method of gene silencing is that certain core promoters are actually excluded from the region next to that red nuclear membrane. And in that exclusion zone are some of the genes. And so without the promoters, the genes can't express. So this spatial segregation is one means of regulating expression. And at the bottom is Harold's work with Claire Waterman at NIH, where he was looking at focal adhesions, the points of contact to the extracellular matrix. Harold created an incredibly sensitive 3D version of Palm, where the vertical resolution is actually even better than the lateral. And with that, they were able to deduce the total vertical architecture of the 90 different proteins that go into these focal adhesion contacts. So all of that is fine, but probably the single application that's had the most biological utility out of Palm so far is something we call SPT Palm. So in this, you're not trying to get very super high density, but you're looking at the diffusion of molecules that you photoactivate in different subsets. So this is a very recent example from my colleague James Liu at Genelia, where he's looking at the proteins involved in Huntington's disease. And so that protein can undergo a mutation with a lot of these polyglutamine repeats, and then that forms aggregates as these proteins clump together, as you can see it right. And what happens is that these aggregates act as traps, not just for the non-mutant Huntington protein, but also for many transcription factors. And so by acting as sinks of transcription factors, they downregulate gene expression for certain genes, and then that has these downstream consequences for the disease. So by 2008, now superresolution is on everybody's lips. Everybody's getting into it. And I was just as disillusioned with it as I was of near field in '94, and I felt like I was living the same bad nightmare all over again. Not only because of the bullshit level going to infinity again, but also because of the inherent limitations of super-resolution fluorescence microscopy. And the first and most important problem is that, remember, you're only looking at fluorescent labels, and these are indirect reporters of the underlying proteins you care about. And if you don't have enough fluorescent proteins decorating often enough, you're going to end up getting just a very patchy, boop, boop. It's like throwing out 99% of the pixels in your HD TV and expecting to get a good image, okay? So it turns out, if you think in terms of Nyquist criteria, that if I want resolution X, I have to sample every X over 2, it means you have to have ridiculously high densities, okay? It's possible to achieve, but it's generally way beyond what biologists have traditionally had to do for their diffraction-limited imaging, and puts all the onus on them, which is hard, rather than on the microscope people to get a good image. Another problem is you have a fork in the road. You can either do live imaging or fixed imaging. So in fixed, you put in some chemical that kind of gives rigor mortis to the cell. It basically looks like roadkill then, and then you image that. But of course, if you're putting this crap in, you're going to change the ultrastructure. What you're seeing it on the right there is the first picture on the left is showing a live cell of an organelle called the endoplasmic reticulum. And then on the right, you end up putting in this stuff, and you can see how it aggregates and so forth. So even before your microscope looks at the sample has been changed. And 95 or 99% of what we've supposedly learned by superresolution is from chemically fixed cells. And I think you need to put a big asterisk next to all of that. The other fork in the road is to do live imaging. But here are the two methods that shared the Nobel Prize: the Stater-Resolve technique developed by my co-laureate Stefan Hell, and then the localization methods developed by myself and W. Morner. Now, the problem with these methods is they require hundreds to thousands to tens of thousands times as much light from the specimen per image as a diffraction-limited image to get the same SNR. They require intensities anywhere from 10 to the 4th to 10 to the 10th times more intense than light evolved under. And they require imaging times that are far longer than the rates at which things move inside of cells. And so it's really extremely difficult to do live imaging by these methods. And it should be obvious, right? And when you think about it for a minute, is that if I want higher spatial resolution, I need to have more pixels. If I need to have more pixels, it's going to means more measurements. That means it's going to take more time, and means I'm throwing more damaging light at the specimen while I'm doing it. So there's always trade-offs between spatial resolution, speed, toxicity, and how deep I can image. So at some point, when I was again thoroughly disgusted with the way things were headed and how everybody's poking up that direction, I realized that's a mistake, okay? And from 2008 on, the focus of my group has been to find any place inside of that tetrahedron which has not yet been served by microscopy developers to service biologists, and try to develop tools in those missing spaces to create new tools that the biologists can learn new things they couldn't learn with previous methods. So one guy who understood this better than any of us was my former colleague Mats Gustafsson, who developed a different super-resolution method called SIM, in which instead of illuminating your sample uniformly, you put on a grading pattern, and that grading pattern demodulates spatial frequencies up to twice beyond the diffraction limit so you can see them. So that's the weakness of SIM, and that was the reason the Nobel Committee said it shouldn't share in the Nobel Prize, is it doesn't have theoretically unlimited resolution, but only a factor of 2. But honest to God, that limitation of SIM is its strength. Because first, you don't need such insane labeling densities. Second, the intensities are orders of magnitude less than any other method. And third, the speed is orders of magnitude faster than the other method. So it's the one method which is truly live cell compatible. You can see that in two examples at the left. Again, the endoplasmic reticulum. On the left, we're looking at it for 1,800 time points, which is 2 orders of magnitude more than the other methods can achieve. And on the right, you're seeing that we're imaging it at 50 frames a second, which again is at least an order of magnitude faster than the other methods. And so, the benefits of SIM for live imaging are huge. And yes, the resolution is only 100 nanometers, but look at the wealth of information you have by having the dynamics along as well. Okay, so it's really quite impactful. The sad news is that Motz, he came to Janelia 2008, and he died of a glioblastoma in 2011. So basically, I inherited his group and have been trying to extend his legacy in this sense. So we offer various flavors of SIM, but the one that most people gravitate to when they come to work with us is something called turf SIM, where you can bring in the illumination at a very oblique angle so you illuminate only the very bottom of the cell. So this prevents any photo damage to the other parts of the cell. It gives really good signal to noise. It means you only can look at the bottom of the cell, but it's still powerful. So here you're looking at a comparison of a diffraction-limited state-of-the-art microscope on the left of a T cell like would fight infection that's been plopped against a cover slip to provoke what's known as the immunological response, what this would do if it were actually attaching itself to a target cell. And so you can see what's happening on the left, but you can see the additional information that you get just with 2x resolution on the right with the turf SIM. So we've used that, for example, with about 25 different groups so far. Here's a couple examples with John Hammer's group at NIH. I'm going to start this movie again so you can see it. So one thing that they're interested in studying is myosin, which is a protein which is involved along with actin in making this sort of structure, sort of like your muscles, the sarcomeres in your muscles, which contract and create forces. And so what happens in myosin is that they form bipolar filaments where single elements come together and create sort of a two double-headed structure you see there. You can see one growing, but you can see when the next one comes, it actually splits off from the first. It isn't actually growing de novo. And now we'll go again, and you see, whoop, there's the next one, and it split off from the original. So that was a surprise in doing this live imaging. And if you look at this movie, you can see, boom, one acts as sort of a cascade, a nucleation center from which all the others, sort of avalanche in production. And so what we learned from this is that while de novo nucleation of these bipolar filaments in the cell is possible, by far a much more energetically favorable thing is for the initial bipolar filament to act as a template for other ones to form from it. We also discovered that there are not just one type of myosin, there are zillions of types of myosins. And in particular, Jordan was able to show that myosin 18 can actually form bipolar filaments along with myosin 2, and by doing so, it's able to modulate the length of these bipolar filaments so that they can be used in different contexts inside of the cell. And the myosin 18 also provides different types of anchors to adapt to different types of targets in the cell. So by being able to mix these two filaments, it adds a lot of flexibility in what the cell can do with myosins when they generate forces. So if the knock against SIM is that it only offers 100 nanometer resolution, a focus of my group is how can we prove the Nobel Committee wrong and show that SIM can do just as well as the other methods from a resolution perspective. So the simple thing to do is to just increase the numerical aperture of the lens to whatever is practically possible. So it turns out there's a turf objective that runs at 1.7 NA, for which case in the green channel we can get down to 84 nanometer resolution, which is pretty darn good. As you can see here, this is looking at the focal adhesions I described before, but now we're looking in two colors in a live cell at the distribution, showing that there's a difference in the distribution between the periphery and the interior between these two proteins. And there's retrograde flow at the periphery of the cell. The example at right is showing the myosin bipolar filaments again, but now also with the actin I described, where they work in concert to create these forces that squeeze the cell that you can see there. One of the many advantages of SIM is that unlike STED and unlike PALM, is it doesn't require crazy photo-switching properties of the molecules, and so you can use any off-the-shelf fluorophores, which is a big plus to the biologists. So we've been using that high-NA SIM to understand, with Tommy Kirkhausen's group at Harvard, how cargoes are brought past the plasma membrane into the cell. So one of the major mechanisms to do that is something called clathrin-mediated endocytosis, where this protein forms this sort of basket that pinches the membrane and then has a cargo from the outside that gets captured and then pulled into the cell by doing that. In the turf mode, we only illuminate the very top of that cage, so therefore it looks like a ring when you go to the turf SIM resolution. And so now that we can measure the diameter of those pits, we can actually then correlate the lifetime of the pits, because we can follow them over hundreds of time points to their different sizes and then see that for the first time. We can also, because we can do multicolor imaging, again, look at the role of actin, the cytoskeleton protein, in clathrin-mediated endocytosis, because there's a lot of debate in the field about, does actin have a role? And so what we found, at least in these cells, is that just prior to endocytosis, actin is recruited to the pit. And when it is recruited, there's a small but statistically significant shortening of the lifetime. We also found that there are many actin rings inside of the cell, similar to the clathrin rings. And we thought at first that these were probably co-localized and being used to generate forces to help pull those cargoes in the cell. Turns out almost none of them are co-localized with the clathrin, and it's still a mystery as to what their role is. So that took us to 84 nanometer resolution, but what if we really want to compete with the other methods? Well, then we need to use the same photo switching tricks that the other methods use. And in our case, what Motz was doing when he died was actually taking a photo-switchable fluorescent protein, which would first turn on all of the molecules, then use a standing wave of excitation to turn off all of the molecules except the ones at the nodes of the standing wave. That would leave little peaks of residual molecules that would have higher Fourier components to demodulate even higher frequencies beyond the diffraction limit. And his grad student Hesper was able to then publish a paper where she was able to push this approach to 50 nanometer resolution, but at 900 seconds per image, so not exactly live cell compatible. So I hired a postdoc, Dong, and we tried to think about how can we make this better, make it live cell compatible and fast and non-invasive. And what we realized is that this principle of saturated depletion, which is basically the same principle behind the STED and resolved approach that Stefan created, is really not a good way to do live imaging. Because first you turn on all of the molecules, and then you immediately turn right around and turn almost all of them off, except for the few at the nodes. Well, every time you make a transition, there's a chance you're going to blow apart that molecule from light, and that's going to create damage in the cell, and it's going to take time. And energy and time are your enemies when it comes to live imaging. So we figured a better approach would be to use patterned activation, where we use one standing wave to turn on a subset of the molecules, and another one of a different color to turn them off and to read out the fluorescence. And so by doing that, we limit ourselves to only a factor of 3 beyond the diffraction limit, but that's still 62 nanometers, and that's pretty darn good. If you can see from this movie, as we scroll from method to method, that we can get quite competitive resolution to the other methods at this level. In fact, here is that competitive comparison. So remember, the Nobel Committee said SIM doesn't belong because it doesn't get as good resolution. But here we're comparing the different methods. At upper left, you're seeing, on left, probably the best image I've seen by localization microscopy of the actin cytoskeleton inside of a cell. And you can compare that, but it's on a fixed cell. And it took 1,600 seconds to get that one image. Whereas the image on the right was done in a little over 1 second and on a live cell, and it's just one frame from a movie of about 30-odd frames, okay? But it was done, therefore, 600 times faster, and it took 20 times less light in order to do. And yet you can look at those images in frequency space, you can look at them in real space. There's not really a dime's worth of difference between them in resolution, despite the claim of the other being 3 times better. And that's an aside I don't really have time for, but in my opinion, the way people have used metrics for evaluating resolution in super resolution microscopy is shameful. It's really not appropriate. And in fact, if you want to have an amusing hour, in late April, after this paper came out, Stefan Hell's group published a rebuttal of sorts in terms of a technical comment on our paper in Science in late April, and we filed a response. And I think you'd probably be amused by both of those if you read them. The next comparison is again to Stefan's resolve technique. So this is looking at caveolae, which is another form of endocytosis. So it should form rings. And so you can see the rings in the nonlinear SIM image, but you can't see the rings in the resolved image despite the claim of higher resolution. At the bottom, you're looking at another cytoskeletal protein, keratin. You can see the resolved image is really not that different from a diffraction-limited image if you compare them side by side. And just even the regular garden variety SIM is clearly better, and the nonlinear SIM is better still. And so, again, be very skeptical of resolution claims in this business. So all of that's great, but these techniques still require turf, and so that limits us to the very 100 nanometers or so from the cover slip, which is pretty damn limiting. Same limit that we had in the near field days. And so how can we do deeper? Well, if you go just outside of turf, you can create sort of a slab of light that's about 3/4 of a micron thick. And so with that, it's easy to do fast imaging. So here's 3 different colors all captured in a ½ second total, and looking at 3 different things happening in the cell at once. The endoplasmic reticulum again in purple, the microtubules of the cytoskeleton in green, and the little yellow bags are lysosomes, these protein-degrading pockets in the cell. And so you can see there's different kinds of lysosomes. There are little skinny guys, like you can see in the still at left, which are under active transport and actually switch tracks as they go between microtubules. The microtubules can slide through the ER and disrupt the ER, and likewise the ER can disrupt at least the shape of the microtubules. So we've been using that grazing incidence mode with, again, Jennifer's group. Jennifer has now come to Janelia as a group leader. So it's great to have an actual cell biologist there to work with us. And so one of the things she's interested in for years is the endoplasmic reticulum. And in most of the cell away from the nucleus, the endoplasmic reticulum looks like a net, as you saw in those earlier pictures. But it has both, little lines of strings, but it also has these sort of fuzzy, large sheets inside of it. But when we look at those with the higher resolution of SIM, what we see is that these putative sheets are really kind of riddled full of holes. And in fact, if instead you look at it live, you see that those sheets are actually riddled with holes, and they're really like little tubules that kind of come apart, reassemble. There's a huge dynamic process going on inside of these things. And there's no way that you can see them with say, a slower microscope, because even at 4 frames a second, it just averages out. So again, what I always say in my talks is that for any microscope, life is a 4-dimensional process, X, Y, Z, T. And you're only as strong as the weakest dimension of your microscope, because you're going to smear your information out in that dimension. And so one of the main advantages of SIM is this great combination of decent spatial resolution with good temporal resolution. And so what they've learned is that basically these sheets are basically clustered trijunctions. So one of the Achilles heels of SIM, as nice as it is because you're taking a series of wide field images and then doing a Fourier space reconstruction, is that you end up getting defeated by out of focus light when your sample is more than a few microns thick. It's just big fuzz everywhere, and you can't even see the little grading pattern that you put on. But if you work in this grazing incidence mode, you're only illuminating the bottom, so it doesn't matter how thick the thing is. So this is looking at a whole Drosophila, or fruit fly embryo, that's about 200 microns in diameter, but looking at the bottom of it. And halfway through embryogenesis, the epithelial cells that you see in that little cartoon at top come up and start to enclose upon these earlier cells called the amnioserosa. And the amnioserosa in this stage undergoes these positive feedback oscillations of expansion and contraction. And you can see here that we have the resolution to see the single actin filaments pulsing inside of those cells as they're doing this. The one on the right, you can see this band at the top and at the bottom coming together. That's the epithelium coming up to close in on these cells. So that's what's called the pursestring structure. And the epithelial cells have these filopodia, those long sticks that have this VASP protein on the end, but the filopodia and the amnioserosa do not have VASP on them. So all of that's good and takes us a little bit away from the cover slip, but as I said before, you know, the cell has to be looked at in 4 dimensions because every living thing is defined by the fact that it's animate, right? So you need that time dimension. And so while we're always going to be able to learn a lot from things like EM and so forth, the only way we're going to really understand how we go from inanimate molecules that are a few nanometers big that somehow come together in some stochastic way and create something which is robust and animate, which we call the cell. It's one of the central mysteries of science. And yet we need high enough resolution across all 4 dimensions of space-time to be able to address that. So how do we get there? Well, the good news is that the standard tools people were doing for live imaging up until recently left a lot of room for improvement. They're basically the wide field microscope, which blasts away at the whole sample, or the confocal microscope, which focuses the light to a point and then collects the light back. But in both of those methods, you're bathing the entire 3D thickness of the specimen, even though only that dotted focal plane is in focus. And so you either have a lot of background or you strip it out, but if you strip it out, you're still bleaching and doing damage to those other areas. So in my opinion, one of the most important innovations in microscopy in the last 10 years was when Ernst Stelzer at the EMBL reintroduced a 100-year-old idea called plane illumination. So in this case, instead of bringing the light from above and back through the same path, is you take a second lens perpendicular to the first, in this case a cylindrical lens, to create a sheet of light. And that sheet of light only illuminates the region near the focal plane, so there's no bleaching and no toxicity of the regions above and below at all. And you don't need to scan a point around or a focus because the whole plane is illuminated. You snap that with a fast camera, then you go to the next plane and the next, and boom, you get your 3D image. So this has been transformative for understanding embryogenesis at single-cell resolution. But my interest was really to try to be able to look inside single cells with subcellular resolution, and their light sheet has a problem because there's a basic trade-off between how flat of a light sheet you can make and how thick it has to be. And over the dimensions of a cell, it means it's going to be about 3 microns thick, but a cultured cell might be 6 microns thick, so you don't gain a lot. So the trick is to use a physics trick, which is at bottom left up there is a Bessel beam. Is instead of illuminating that side lens with a round spot, you illuminate it with a ring. And by changing the diameter of the ring, you change the cross-sectional thing of the light that comes out. And by changing the thickness of the ring, you change how long the spear of light is that comes out. So you have independent knobs, 2 knobs to tweak. And so that's basically the same technology which is used to read barcodes in supermarket checkout scanners, because then they have a very no loss of focus as that beam is coming across. So if you use that as a little pencil of light and bring that in and out of the plane, then you can make a sub-micron thick light sheet. That way, take your image, move up plane by plane, and get your image. There is one problem with the Bessel approach, which is a little hard to see on this monitor, but a Bessel beam isn't just a little spot of light. In cross-section, it looks like a bullseye. And these side lobes create some out-of-focus excitation when you sweep the beam along to create a little bit of damage, bleaching, and also retard the resolution a bit. So the solution we came up with was, instead of sweeping the beam continuously, we would step it discretely. And then that would create a grading pattern inside of the sample. And then we could make lemonade out of our lemons of these side lobes and basically then use the sort of 2-dimensional grading pattern that resulted from stepping it to basically use Motz's principles of structured illumination and then do mild super resolution with that. And so what you're seeing here now is then that mode, this structured illumination mode, where we're looking at a cell that's been transfected with a cancer signaling protein that promotes these membrane ruffles that fold over and encapsulate extracellular fluid in a process called macropinocytosis to create those little bubbles that you see in that X-ray view at lower left there. Now, so we're on a good path here. This is a good trajectory. The one thing that we learned, though, is that it was too darn slow to step the beam like this. And so what we did is we put in a diffractive element to create an array of 7 parallel Bessel beams so that each one would only have to step over 1/7 the field of view. And the real shock to me in doing this, and probably the most important observation I've made, I think, in my career, was how much less toxic it was to run with 7 beams instead of 1. And what I've learned in that time is that while the total dose of light that you throw on a living specimen is important, a far more important metric of its health is the instantaneous power that you deliver in the cell. So seen in this light, that confocal microscope I described earlier with this focus, which is the gold standard of live imaging, is probably the stupidest possible way to do live imaging. Because not only do you have these cones of light above and below the focus creating unnecessary damage and bleaching, at the focus itself you have an actinic spot of light that's going, and it's leaving death and destruction in its wake as it goes along. So the moral of the story is a line is better than a point and a plane is better than a line. So why the heck would you stop at 7 Bessel beams? Well, the concern was what would happen when the Bessel beams get close enough that the side lobes appreciably interfere, because we were worried of having negative consequences like you see in upper right there, where you have sort of constructive interference and crazy patterns. And sure enough, you do get such things, but there are also other patterns like you see at the bottom 2 examples here, where everything works to your advantage, that you get near perfect destructive interference of the side lobes outside of the plane. So that gets rid of the side lobe problem for you. You have 100% modulation of the intensity in the plane, which is ideal for applying structured illumination. And you spread the energy out over the entire plane to absolutely minimize the peak power, which is great for minimizing phototoxicity. So I'm an engineer, and I always think in terms of trade-offs. I never believe you get something for nothing. So when you get 3 things happen to your benefit in the same thing, it's like finding a unicorn, okay? You feed it carrots and you take it home because it's a big deal. And so that's what we now call, and this is actually derivative of the idea that I was working on that I abandoned when Harold and I got the idea for Palm. And we call this the Lattice light sheet microscope. So that's showing a lattice light sheet, and this is going through, the cell is swept through that light sheet. The orange represents the fluorescence at the particular plane where the light sheet is, and you can just go very quickly and non-invasively through lots of things. So here we're looking at upper left at a neutrophil cell, which is now actually migrating through a collagen mesh. A lot of what we know about cell motility is by looking in a 2-dimensional system, and now we have the ability to look at motility in 3D. At lower left here, you're looking at tetrahymena, which is a model organism, a pond protozoan. He's trapped in a little pocket of agarose, and he's trying to frantically to find his way out of there. But you see that we're looking at that thing for over 1,000 volumes, and he's still okay, and he's still doing his best to get out of there at 3 volumes a second. So both the toxicity measures and the speed measures blow away any other live cell imaging technology by at least an order of magnitude. At upper right there, you're seeing Dictystelia, which is another model organism, a slime mold amoeboid, and this is looking at an actin-associated protein, but you can see in a second, you see that little lightning bolt. So when we render that movie, our collaborators say, "What the hell was that?" And we said, "We don't know. You're the biologist. You tell us." You know, and we've had 70 different groups come to use this technology in my lab and another 40 in our advanced imaging center now. And everybody goes away with 10 terabytes of data and big smiles on their faces after a week. And the smiles quickly fade when they realize they have no idea how to even handle 10 terabytes of data. And that is the Achilles heel of this technique, is there's so much unmined data inside of there, and people just choke on the data that we give them. And it's very frustrating, but that's really the problem with this method. But it reveals so much just on a descriptive level. I've never felt more like Galileo than I have with this microscope, because everybody gets so damned excited when they see the first data, because they've never seen cells with this combination of space and time resolution before. So if you like these, we have a Vimeo album with like 40 different examples if you want to see pretty pictures of wiggling cells. So again, to further emphasize the speed and low toxicity, this is looking at a field of dividing cells and looking in 3 different colors at once at chromosomes, mitochondria, and the endoplasmic reticulum, and then computationally slicing the 3D data in these 2-micron thick slabs. If you look at upper right, there's 1 slab. What you can see is that the mitochondria fragment during cell division. The endoplasmic reticulum goes from a net to folding up into these little pockets. The mitochondria fit inside of those pockets, and they're carried along as the cell divides. So there's just a lot of direct observations you can do even without doing significant analysis. But the reason I show this is that cells are notorious for being incredibly light sensitive during mitosis. They don't want to create replication errors, so they have these mitotic checkpoints where they'll shut down if they're unhappy. These cells did not shut down, despite the fact that that movie represents over 300,000 2D images of the same field of cells. So one of the other things the method is good for is doing single molecule imaging, because the light sheet is so thin that normally one of the big problems with single molecule imaging is, again, out of focus light. So this 35 micron spheroid, you're basically sunk in looking at single molecules by wide field at the bottom. But at the top there, if you park the light sheet at one plane, only the molecules in focus are illuminated. So it's like having that turf mode, but now anywhere in 3D. And so we've used that to look at the binding kinetics of transcription factors to DNA. And by looking at differences in residency time, we've been able to find out where the hotspots of transcription are. So we've been able to do that for super resolution, but I'm going to just skip over that because I'm already at yellow and I have lots to say. And this is looking at dead stuff again. And I swore after 2008 I'd never look at dead stuff, but I got pulled into this because this was the solution to the problem of labeling density. So I can talk about it later if you'd like. In fact, I'm going to skip over some of this too. I want to skip to the end. So there's lots and lots and lots of applications you can do of Lattice, and I will skip over all of those because I want to spend the last couple minutes on something else that I'm always most interested in what I'm doing now, right? So we've taken the Lattice for cells, for cell-cell interactions, small embryos, but then we hit a wall. And the wall is the fact that when you go into a multicellular system, it has to have different refractive indices for the different components inside of it. And so the light sheet gets scrambled going in. And likewise, even if you had the light sheet going in, the light coming out is scrambled on its path to the microscope objective, just like the water on your windshield. And so we need to get around that problem to be able to go deeper into whole specimens. So this is a problem astronomers had to face 50 years ago because the atmosphere does exactly the same thing. Their solution is adaptive optics. They shine a laser into the stratosphere to excite sodium atoms to create a bright artificial guide star next to the magnitude 25 thing that they want to see. The light from both of those things come through the same atmospheric path into the telescope. The guide star light gets split off, goes to a sensor to measure how the wavefront is scrambled, goes into a computer, you change the shape of a deformable mirror to exactly balance those wavefront errors, get back to a good wavefront, and bing, get back to your diffraction-limited imaging. So there's no ground-based telescope on Earth worth its salt of a given size that won't start using adaptive optics. So we do a very similar thing now in our microscopes, where we use 2-photon excitation because it only excites a point instead of a cone inside, in this case, a zebrafish embryo. And so that excites the fluorescence locally. We again use exactly the same type of technology with a trick or two for microscopy. Without the AO, because the light rays don't come to a good focus because they're all scrambled, you get a fuzzy patch. You get low signal and low resolution, but with the AO on, boom, you go back to diffraction-limited imaging and good signal. So these are two examples of how we first developed this in point scanning microscopes. So left, we're looking at a sparse subset of neurons in a zebrafish by point scanning 2-photon microscopy. And the example on the right is doing point scanning confocal microscopy because it's easier to do multicolor imaging there. So one of the challenges in doing it in microscopy is that you're going through different tissue as you go to different parts of the specimen. So this required 20,000 different adaptive optic corrections in order to do. We're going 250 microns deep. We turn the AO off. That's what you would see with a state-of-the-art 2-photon microscope without adaptive optics, and that's what you get with. So many biologists like to believe when they're doing multicellular imaging, they're getting diffraction-limited imaging, but I'm here to tell them that they're wrong, okay? The example on the right, you can see the mitochondria, those purple worms inside of the membrane in green there, that we're seeing in a soma of a neuron 150 microns deep, but without the AO, it's just a fuzz. So, so this brings me to, again, the point of my research going forward is that I am not a biologist, but I was trained as a physicist. And in physics, what we learned from Heisenberg is that you cannot observe a system without affecting the system. And it's my belief that we have never seen the cell in its true physiological state for 3 reasons. First is, as powerful as fluorescent proteins have been, until very recently, we've had little control over the amount of fluorescent protein which is created inside of the cell, and so we end up screwing up the homeostatic balance. There are these new genome editing technologies, specifically CRISPR-Cas9, which fixes that problem, so now we get to endogenous levels. Trouble is, endogenous levels are often a lot weaker than what the biologists have been used to, because oftentimes they've driven up expression so they can have lots of signal in their microscopes. Well, that's where lattice light sheet microscopy comes in, because we can deal with lower levels of expression, and we can deal with doing it in a physiological way where we're not putting in so much light that we end up disturbing the physiology of the cell as we're measuring it. But there's a final challenge, which is that too much of what we know about cell biology has come from studying cells on cover slips, but it's damn for sure that they didn't evolve there. The other thing that you know, if you do any biology for any length of time, is that the gene expression inside of cells is totally dependent upon the environment in which they're located. And so all of the downstream phenotypes, whether they be structural phenotypes or dynamic phenotypes, are totally dependent on the environment. And so the only way we're going to see the cell in its physiological state is to study it in the native organism in which it evolved. And so for that, we need the adaptive optics. So now we have to pull all of these pieces together, and that's what we've been working on. So this is our combined lattice light sheet adaptive optical microscope, looking at membranes in a zebrafish. That's with the AO on, turning the AO off, going deeper again, and there. And now we'll start to build up a volume. And so when I showed you that volume before by the 2-photon microscope, that was pretty, but it took 18 hours to get that volume. This is a comparable sized volume, but it's done in 7.5 minutes because we're using the lattice instead of the 2-photon. And now we're getting to where we want to be, which is to study the dynamics of cells inside of their native environment. This is using it to look at organoids, which is sort of a halfway house where biologists make sort of artificial multicellular systems. In Matrigel, they have lots of aberration. You turn on the AO, the aberrations go away, and we can study endocytosis again, but now in a multicellular system. On the right, you're actually looking at a real multicellular system, the zebrafish again, with Tommy's group, and looking at clathrin-mediated endocytosis. This is the funnest one, though. That's with the AO off in the ear of a zebrafish. That's turning the AO on, and you can see that. And then there's these dextran particles, and now this is in this perilymphatic space in the ear. There's these neutrophil cells that are cleaning up all the garbage, and so they come in and they pick up all that stuff. Then you can see the volume rendering over there. So in other words, we're finally there. We're finally getting good resolution in 4D and being able to look at cells in their native environment. This is where I always wanted to be, was with this microscope right here. This is another example where we're looking in the eye of the developing zebrafish. This is looking at the membranes. This is looking at different organelles inside the mitochondria and the ER. And you can see how, boom, there was a cell that just divided right there, right, in this sort of cutaway view. The other problem we have is, you know, at this point, again, I told you about the collaborators swimming in data. We have a cluster. We have lots of storage space at Janelia, and we're consuming it all. We can easily take 50 terabytes in a week, often 100 terabytes in a week. And just even the visualization, the porting of the data. You know, how do you deal with a 4D data set? You know, you take little slabs and slices and you're seeing a tiny fraction of what's there. It's a huge problem. Anyway, I'm way out of time, but I want to say two other things. First is that I don't give a crap about papers. I give a crap about impact, okay? And the only way we're going to have an impact is to get these tools in the hands of biologists. And I find that that's a bigger challenge than building the tools themselves. And so we have a three-prong approach at Janelia to doing that. And the first is this Advanced Imaging Center we created with the Gordon Moore Foundation, where we have copies of all the advanced microscopies developed at Janelia in a center which is well-funded, well-staffed, which is only for outside people to come and try out these tools and see if they like them. If they can get enough data for a paper, great. If not, they can at least get hooked on this and start putting pressure on the microscope companies so that the companies realize there's a market big enough for them to take the risk to do the R&D to make a commercial instrument. Because in the end, a commercial instrument is the only way that it's going to get in the hands of many people. Places like the ASC is going to be too narrow a bottleneck in the long run. It's already too narrow a bottleneck. We're totally oversubscribed by about a factor of 6 for time on these microscopes. So that's how to do it. The other part of the solution in the temporary cases, we document our microscopes down to the screws. And so anybody who signs a research license, we give them all that information so that they can build their own. So far, about 80 different groups have executed licenses for the Lattice tech. In terms of running Lattice scopes now, there's probably about 5, but then, you know, it takes a while to get there. So I think they'll get there. The last thing about this dissemination is something new that caught my fancy. This is not my technology. This was developed by Ed Boyden at MIT. The best ideas are crazy ones, and this is one that I thought was completely crazy, which is instead of getting super resolution by optics tricks, what you do is you take your tissue, you infuse it with a polymer, and then you link the fluorescent proteins or other fluorophores to the polymer, and then you digest away all the proteins, all the lipids. So all that's left is the polymer and the fluorophores. And then you change the osmotic concentration of the water, and then the whole thing expands, just like when you pee in a baby diaper. Okay, the whole thing just, and so it expands like 5-fold. And then you image that in your diffraction-limited microscope, and you get your resolution gain because you've actually expanded your sample instead. Sounds nuts. And these, you know, they had a paper in early 2015, this one. I thought the data looked like crap, and I thought I could explain everything in terms of artifacts. And then they wanted to come out and try it on the Lattice in January, and we put their stuff in, and I was just blown away. I mean, just, you know, I mean, it was amazing. So this is what's been scrolling here is the soma of a single neuron, and all of those voids are the mitochondria that you're seeing because they're just excluding the fluorophore that's just floating in the cytosol around the middle of the cell. And you can also see the endoplasmic reticulum by this negative staining, the nuclear membrane, and so forth. And you can cover, with Lattice, you can cover huge volumes very quickly. So this is, again, just looking in the unexpanded state around 90 by 70 by 50. But this is potentially something that could put optical connectomics back on the map to be able to look at huge volumes of tissue very quickly. It also puts a lot of interesting cell biology in the context of the whole tissue again on the map, where you could look at differences due to different cell types and so forth and different organelles. You know, I think the Lattice and the expansion is a really good combination, but again, it's easy to get 50 to 100 terabytes in a week with this stuff, and it's just hard to deal with all of that data. If I have one plea to Simons or anybody who wants to think about this is we need to have people who can good bioinformatics people, guided by good biologists with good questions to be able to extract meaning from this data, or ways of at least visualizing it and manipulating it. And so with that, I'd just like to quit and thank you all for being here. We've had so many collaborators. Like I say, between the AIC and Janelia now we're up to 100 at least, and that's been a huge strength of being at Janelia. The visitor program, to bring in people to come work on these scopes, is a major advantage. The other major advantage, of course, is the 100% focus on research, which those of you who are here at Simons probably can appreciate. But as a result, I can work in a small group, and I really feel like this is my group when it was at its biggest. It's at 4 now and soon to be 3, which is my comfort level. And I feel that it's the fact that our group is so small that allows us to outcompete everybody else. Thanks a lot.